Hi everyone, I'm Julie Fleshman, President and CEO of PanCAN. I'm so excited to be with you today to have this important conversation with leading experts about how we make progress in developing an early detection strategy for pancreatic cancer. In fact, just this week, PanCAN launched a $25 million interventional study with the goal to create a screening process for pancreatic cancer and to change practice guidelines. We call this study PanCAN's Early Detection Initiative, and we believe it has the opportunity to change the way we detect and treat pancreatic cancer. As many of you know, pancreatic cancer is the third leading cause of cancer death in the United States. It's also a difficult disease to treat. One of the reasons for that is because there is no early detection test, but also the symptoms for pancreatic cancer are quite vague. So usually by the time someone is diagnosed, it's late stage and then more difficult to treat at that point. So finding an early detection test that can detect the cancer when it is a patient can still have surgery will be critically important to changing patient outcomes and long-term survival. So we know that there are other cancers that have early detection tests. Breast cancer has a mammogram. Colon cancer has a colonoscopy. So why don't we have a test like that for pancreatic cancer? Today we're going to talk about some of the challenges in developing a test and what we need to do to overcome those challenges. Joining me here today are three special guests that I'm really excited to introduce you to. First, Dr. Lynn Matrisian is PanCAN's Chief Science Officer. Lynn brings extensive cancer research experience and business training to provide strategic direction for PanCAN's scientific initiatives. Before joining PanCAN, Lynn worked for 25 years leading a research laboratory dedicated to understanding cancer metastasis. Next, we have Dr. Rosalie Sears. She's a professor in the Department of Molecular and Medical Genetics, the Krista L. Lake Chair in Cancer Research, and the co-director of the Brendan Colson Center for Pancreatic Care at the Knight Cancer Institute at the Oregon Health and Science University. She's also a member of PanCAN's Scientific and Medical Advisory Board and a past PanCAN grant recipient. And finally, Dr. Brian Wolpin, a medical oncologist and the chief of the Division of Gastrointestinal Oncology and the Robert T. and Judith B. Hale Chair in Pancreatic Cancer at the Dana-Farber Cancer Institute. He's also an associate professor of medicine at Harvard Medical School. Dr. Wolpin is a past PanCAN grant recipient and the PanCAN Precision Promise Principal Investigator at Dana-Farber Cancer Institute. Thank you so much all for being with us today. Before we begin, I want to thank our webinar sponsors, Ipsen and Sylvester Comprehensive Cancer Center. Thank you so much. Without your support, we wouldn't be able to provide critical information like we're doing today. I'd also like to thank our scientific and medical affairs industry members, AbbVie, AstraZeneca, FibroGen, Ipsen, Pfizer, Raphael Pharmaceuticals, Tempest, and Time. None of this would be possible without sponsors like those and without the generosity of our viewers and our donors like you. To help support our mission, please visit pancan.org. All right, so let's get started. So as I mentioned, pancreatic cancer doesn't currently have an early detection test or screening method. And one of the issues that makes this challenging is that if a potential test is developed, you can't validate that it works by testing it on every American. That would be way too expensive. So you need a group of individuals that are at a higher risk for developing pancreatic cancer, and then you need the potential early detection tests so that you can validate to see if it actually works and does detect the pancreatic cancer earlier than it would otherwise. So Lynn, let's talk about this. This can be confusing. There's a lot of steps that are involved. Can you explain this concept of needing both a high-risk group as well as a test, and what's the process for how you validate the test?
So thanks, Julie, and hi, everybody. So think about a test that we could give to virtually everybody in the United States on a regular basis. to find just a few individuals who unfortunately have pancreatic cancer and don't know it. So that test would have to be very easy, very fast, very cheap, and not cause any harm at all. So how would you find something? What kind of test would that be? So we usually think of like a blood test or perhaps a urine test, something like that, the kind of test we get in a doctor's office when we go for our annual physical. So how would you then use that for pancreatic cancer? Well, what scientists have been doing for a while now is they've been taking, say, blood from people with pancreatic cancer and comparing it to those without pancreatic cancer and looking for what we call biomarkers, something in there that distinguishes them, that says this person has pancreatic cancer and this one does not. But then the problem becomes, how do you know whether that's a good test for the early detection of pancreatic cancer? All those individuals we know already have pancreatic cancer. How do we know that test is sensitive enough in order to pick it up before any other symptoms of pancreatic cancer? Well, that's where the high risk group comes in. If we had to do that on the whole population, we would find, say, 13 cases of pancreatic cancer in 100,000 people. So that's 0.01%. So we'd have to look at lots and lots of people to know whether that test was good enough. And there are a few samples like that are blood that was taken from individuals that a couple years later got pancreatic cancer. And I think you'll hear about some of that from our panelists today. But the way to do that much better and much more feasibly is to use a high risk group. Some in a case where perhaps you would screen 100 people and find a case of pancreatic cancer, so a 1% chance. And that's really the value. We need the test and we need the population to screen that test to know whether we're really finding and whether we really have a test that will pick up pancreatic cancer early before those other symptoms that lead to a diagnosis.
Great. Thanks, Lynn. That was a terrific explanation. So let's talk then about some of the high-risk groups that have been identified for pancreatic cancer. Brian, the group we probably hear a lot about is those people that have a genetic predisposition to pancreatic cancer. So can you talk about some of the genetic risk factors and who makes up this high-risk group?
Sure. Thank you, Julie. And I first just want to thank Julie for the invitation and the honor of serving on this panel with Dr. Matrisian and Sears. It's really an honor to do so. So, I think if we ask about genetics, really what we're asking about is the DNA that you inherited from your parents, the genetic code that was encoded from your parents. Does that genetic code have a difference in it compared to others in the population that may predispose you to get pancreatic cancer? And this really has been a very active area of investigation over the past, really in particular, 5 to 10 years. And really with the goal of identifying those individuals in the population whose genetics may suggest that they're at high risk or higher risk of developing the disease. So when we think about this, we usually think about it in two different types of categories. One are very rare genes, so genes that may have a mutation in them or some other change. You'll hear a lot of different terms used sort of interchangeably, mutation or polymorphism or variant. These are all saying the same thing, which is really saying a change in the DNA that you inherited. But there are rare mutations in genes, less than 1% of the population, that may carry a risk for pancreatic cancer that is, say, 3 to 10-fold what it would be in the normal population or the general population. Some of these genes people have heard of, like BRCA1 or BRCA2 or others like CDKN2A. These are all genes that, when they're mutated in this rare setting, confer a higher risk of pancreatic cancer. And in particular, this is a population that we've been thinking about how to screen, right? How do we try to find the cancer early? Because these mutations we know confer this higher risk. I'll take a very brief tangent, but I think one other important thing for us to remember is that now national guidelines have changed and suggest that all patients who present with pancreatic cancer should have genetic testing. I just think this is important for patients to know. And the reason for that is over time, as we've done these studies, we've realized between 5 and 10% of all patients who present with pancreatic cancer will have one of these mutations that was inherited. And that's really important for two reasons. One of which is it may allow us to change the therapy for that patient, meaning if they inherited one of these mutations, we may actually be able to treat that patient differently. And that's really important for their care. But equally as important is something called cascade testing, where if we find a mutation that was inherited in a patient with pancreatic cancer, we can then offer genetic testing to family members. And that ultimately, as Lynn was saying, allows us to define a group that's at higher risk, which we then be able to apply these screening tests to. So it's just a very important thing that if your doctor doesn't bring up to you this genetic testing, that you mention it to them. So these are rare mutations. Less than 1% of the population will have them, and a relatively higher risk, 3 to 10-fold. The second category that we think about are sometimes called common variants. And these are alterations that are actually quite common in the population. So maybe 20, 30% of the population will carry this variant in their DNA, but the risk is very small, right? So the extra risk may be 1.1 or 1.2-fold rather than this 3 to 10-fold. Right now, these have not been clinically actionable. So in clinic, we don't really use these currently, but there is a lot of research going on to think, can you combine these common variants into scores that may allow us in the future to identify individuals who are at higher risk of pancreatic cancer, even if they don't have one of these rare genes like BRCA2 or others. And so I think it's really both of these areas of investigation that have made big steps in the past 5 to 10 years that are trying to identify then individuals in the population who have a higher risk of pancreatic cancer because of their genetics.
Great. Thanks so much. And I think that was, I'm so glad that you reminded everybody about the recommendation now for all patients to get this testing, not only to help them, but also to identify these high-risk groups that we're talking about today. So thank you. So Lynn, one of the more newly identified high-risk groups is that of those individuals with new onset diabetes. Can you tell us about that high-risk group?
Sure. So it's actually, people have known that pancreatic cancer can cause diabetes for a long time, but it really hasn't been recognized as an early symptom. The pancreatic cancer, of course, occurs in the pancreas, and one of the many jobs of the pancreas is to control our blood sugar levels. And so having pancreatic cancer can talk to the cells that regulate your blood sugar levels and somehow disrupt it so that diabetes occurs. So what we now know is that can be in only about 1% of patients be an early symptom of pancreatic cancer. And the problem is that diabetes of this sort is actually quite common in individuals, say over 50 years old or something like that. And so it's really, again, a matter of distinguishing diabetes that's caused by pancreatic cancer from diabetes that is type 2 diabetes, as it occurs in a large part of the population. But because of a lot of research that's been done over the years, and in particular in recent years, we've really recognized that this is a high-risk population. 1 in 100 individuals with new onset diabetes has a risk of pancreatic cancer for the next 3 years. And that gives us an opportunity to take advantage of that knowledge and screen that population and see if we can find an early detection test for pancreatic cancer.
Great. Thank you, Lynn. So Rosie, how about the high-risk group who have cysts or IPMNs?
Great. Thank you for the question, Julie. And I want to echo what Brian said and tell you what an honor it is to serve on this panel and how much I appreciate the opportunity to share with the audience some of our thoughts on the early detection of pancreatic cancer. So you asked me about cysts and IPMNs. So just to back up a little bit, pancreatic cysts are small growths that can occur in the pancreas, and they're fluid filled. And they actually are relatively common in older individuals. They're usually found in routine imaging that's being done for other reasons. And most of them are not cancerous. And yet they are a bit worrisome and they do require follow-up. And so it would be important to have follow-up imaging, including CT scans or MRI imaging. So these are clinical imaging modalities that your doctor can order. And what the doctors would be looking for is any kind of change in these cysts or lesions that they see in the pancreas. And so if it grew bigger or if it looked like it had any kind of solid part to it, the doctors would send you for an endoscopic ultrasound, which could then be followed by a biopsy. So patients that have these cysts or IPMNs, so to speak, which stands for intrapapillary mucinous neoplasias, these can be precursors for pancreatic cancer. And that's why it's particularly worrisome when these kind of cysts, which can occur in other organs in your body, occur in the pancreas. And so patients generally with these cysts, particularly if they have mucinous cystic neoplasias, those patients are often sent to surgery pretty immediately because there's a high chance that those could be cancerous. Patients with IPMNs are divided into two groups, those with main duct IPMNs. So that would be a neoplasia in the main duct of the pancreas. And those are more worrisome for pancreatic cancer and will be typically biopsied and go on, and those patients would often go on to surgery. For IPMNs, they can also be in the branching ducts of the pancreas. And in that case, they're often not cancerous and they're benign. And so you wouldn't want to subject somebody to the risk of a pancreatic surgery in that case. And so these patients are placed in surveillance, and that means that typically they're imaged again with a CT or an MRI annually. There are some ongoing clinical trials actually, where they're testing whether it would make a difference to image patients every 6 months rather than every year, for example. And again, those images, the imaging would be followed up by an endoscopic ultrasound and biopsy if any kind of worrisome features are found. The other interesting thing about patients that are being followed in high-risk clinics that have IPMNs or other types of cysts is that actually the fluid from the cysts can be collected, and then they could be subject to different types of testing, like DNA testing for mutations that Dr. Wolpin referenced. And they can also be tested for protein biomarkers. So this is something that some scientists and researchers have been interested in, collecting these cyst fluids for doing detection tests on. So I think you were also asked me about pancreatitis. Chronic pancreatitis is another risk factor, particularly if it's hereditary, which would be the case as Dr. Wolpin was referring to. There's certain genes that are known to cause pancreatic cancer. There's genes that can cause pancreatitis, and those can also raise your risk factor for pancreatic cancer. So patients with chronic pancreatitis are also frequently in high-risk clinics and screening protocols, again, where any kind of worrisome features would be followed up with an EUS-guided biopsy.
Great. Thanks so much. So that was a terrific overview of some of the most common high-risk groups for pancreatic cancer. So the second step after we identify this high-risk group is a screening test needs to be developed. So let's talk about some of the screening tests that are currently being developed. Rosie, I'll start with you. There's a pancreatic cancer-specific blood test that's being developed right now from a company called Immunovia. Can you talk about that test?
Yeah, I'd be happy to. We're pretty excited about this possibility as an early detection test. So as Julie mentioned, and also as Lynn mentioned, being able to sample the blood is a relatively non-invasive way of that generally has no risk associated with it, to look for kind of markers that could be in the blood that would indicate that you had pancreatic cancer. And markers that scientists have been mostly focused on include proteins that can be floating in your blood, DNA that can be also floating in your blood, and these can be assayed. The Immunovia test in particular is focused on proteins that float in your blood. And they're based on their name, Immunovia, they're particularly interested in the immune system and how your immune system would be reacting to having pancreatic cancer. And so they're trying to monitor for proteins that float around in your blood that are there to activate the immune system. And so what they did is they generated a small little chip that is coated with antibodies and those antibodies are specific for recognizing the proteins that your immune system would be giving off if it thought you had cancer. And then those proteins become labeled, and a scanner can detect whether you have those proteins in your blood. The protein, so they started with a very large number of antibodies to detect over 500 of these immune-related proteins that could be floating in your blood. And they used what Dr. Matrisian was referring to, retrospective blood samples, which were blood samples that had already been collected from patients with pancreatic cancer or healthy patients. And they were able to get the 500 proteins down to only about 30. And with that, they were able to show that they had a high sensitivity and specificity for detecting patients that had pancreatic cancer or not. But again, as Dr. Matrisian mentioned, this is a little easier than saying, can we detect a patient that has very early stage pancreatic cancer, or even before they could develop detectable pancreatic cancer based on body imaging, for example. And so the Immunovia company refined their biomarkers, and they call this panel called PanCAN D is their biomarkers, and they've got it down now to only 8 antibodies, but they have added to it CA19-9, which is a commonly used clinical blood protein for detecting pancreatic cancer. And when they do that, they're actually able to detect early stage pancreatic cancer, which would be stage 1 and 2, versus not just healthy controls, but patients with diabetes, or even patients with symptomatic, like where patients come in because they're having stomach or lower abdomen pain, or different reasons that they've come in with suspecting that something's wrong. And they've been able to get a very high sensitivity and specificity for detecting stage 1, 2 cancer from even these symptomatic or diabetic control patients. And so they're currently running a study, a clinical study called PanFAM-1. And they focused, again, as Dr. Matrisian mentioned, you need to focus on high-risk patients because screening the general public is very difficult in pancreatic cancer. And so the PanFAM study is actually focused on patients with familial or hereditary pancreatic cancer, as Dr. Wolpin mentioned. These are patients with genetic mutations, known genetic mutations that would be likely to give you a higher risk of pancreatic cancer. It doesn't mean you're going to get pancreatic cancer. You just have a higher risk. And so their goal was to collect 3,000 patients' blood samples. And they've been working on this now for several years. There's 23 participating sites, most of them in the United States, but some in Europe. And they're on target now for finishing the 3,000 sample collection by April 2021. And then the second half of the study, what they're going to do is they're going to evaluate how all the tests went. Nothing has changed in terms of how these patients are being studied in their high-risk clinics. But what they're going to do is they're going to evaluate whether their test worked and has high enough sensitivity and specificity to then guide a second half of the study, which would be considered interventional, which means that in this case, if a patient has a blood sample and it tests positive on the Immunovia test, then they would be sent for an additional imaging scan. So we're excited about this study and how it will work out.
Great, thank you. So yeah, you really shared with us how you take that test and then how you study it and validate it in a high-risk group. So Brian, you've been working on tests for pancreatic cancer for many years. Can you explain what a biomarker is, and then what are you finding? What are you finding are promising biomarkers?
Sure. Thanks, Julie. So I would say biomarker really stands for a biological marker, right? That's what biomarker means. And it's really a marker that you use to try to understand the biology in a patient, right? It has a few characteristics that are important. One is it's something you measure, right? So it's not something that I ask my patient in clinic and they respond, or it's not something when I'm doing an exam I can feel. This is really something you measure. The second is it's something you measure in a body fluid or tissue of your patient. So examples of that would be in the blood, as has been mentioned before, as we've been talking today, or urine, or you take tissue from a patient, you look at the DNA changes in a tumor. These are all biologic samples from a patient. So measured, a biologic sample, and then related in some way to the biology or physiology of the patient. Right, so I'll give you a few examples, sort of illustrate some of what a biomarker could be, right? So a patient comes into the emergency room and they have chest pain, right? So we would measure something in the blood called troponin. And if that was high, it would tell us right now, when we're seeing this patient, this biomarker is telling us the patient is having a heart attack, right? That's a biomarker that informs you right now about the diagnosis of a disease. Another type of biomarker is something like cholesterol, right? You go to your primary care doctor, they take a cholesterol level from your blood. They're not asking, do you have a heart attack now? They're asking, can we predict in the future who's at risk for having a heart attack? And that's also a biomarker, right, in this same sense. And then a third example would be, a patient comes to see me in clinic and we sequence the DNA in that patient's tumor. And then we then try to choose the therapy for that patient based on the mutation in that patient's tumor. In each of these cases, these are things we've measured. We used a patient's sample, blood or tumor, and we've used that to inform us about the biology in that patient. So if we think about what are promising biomarkers for pancreatic cancer, we would usually divide them into two categories. So one would be a risk biomarker, and the other might be a detection biomarker. So a risk biomarker we've just been talking about, actually. So again, to go back to the example where we took someone's DNA and we asked, did they have a mutation they inherited in that DNA to tell us that they had an inherited form of pancreatic cancer that therefore would put them at risk for pancreatic cancer in the future? That's a risk biomarker, right? We've checked something, we then look forward and we say they're at risk for developing this disease. Another, which is what Rosie was just talking about, is a detection biomarker, right? And this is something where we measure the marker and then we're looking and trying to see, do they have the cancer now, but we just don't know it yet, right? And that's really trying to find the cancer either when it's asymptomatic or find it early, meaning, well, they're having some symptoms, but we don't know what's going on. And let's try to make this diagnosis earlier. So it's really trying to speed up the diagnosis of their pancreatic cancer. And I think as Rosie mentioned, there really are a number of biomarkers that are under study that are moving towards the clinic, right, that are being tested in patients. One of them, which Rosie also mentioned, was looking at fragments of DNA that are present in the blood, right? So it turns out that all of the cells in our body will shed some of their DNA, and that DNA ends up in the bloodstream. And you can measure that DNA by taking a blood sample and looking at it. If someone has a cancer, some of the cancer's DNA also ends up in the blood. And then the trick becomes, can you differentiate what is a DNA sample or a DNA fragment coming from a tumor versus all the other DNA that's being spilled into the blood from their normal cells? And there's now been a lot of work in this area trying to see, can we find a mutation in the blood that might have come from a cancer? Or can we find a methylation difference? These are little marks on the DNA that tell a gene whether to be turned on or off? Or can we see different characteristics of those DNA fragments in their structure that might tell us that they're coming from a cancer and not from normal cells? And there, again, over the past 5 or 10 years have been some major advances in these areas, now trying to use these DNA fragments to be a little flag or a signal to us that there's a cancer here and we just don't know it yet. A second type of biomarker that's making its way toward the clinic, which Rosie mentioned, are protein markers, right? These are things that are secreted from the tumor that end up in the bloodstream that, again, we can try to measure. And so CA19-9 is sort of the quintessential protein marker. Right? Many people have heard of it because we use it in the clinic to actually follow patients with pancreatic cancer who are on treatment. But it turns out it may also have some utility to try to find the cancer earlier. And then you can take other proteins and add those to CA19-9 and try to come up with a panel of different proteins that may be related to a cancer that's present, that we can then try to find those people among these risk groups. And then there are a variety of others along these same lines, or this same principle, which is tumor cells are different than normal cells, and they may secrete things into the bloodstream that we can find. And you may come across, there are studies looking at fragments of RNA. There are studies looking at little vesicles called exosomes. Really, though, the principle behind many of these is very similar. It's trying to find these little fragments or parts of the tumor cell that end up in the blood. I think another important type of biomarker, in particular detection biomarker, also is something that Rosie mentioned. And that's saying, let's not only rely on things that are secreted or put into the bloodstream from the tumor, but can we also use the body's response to the tumor, right? So when an early tumor is in place, the body does know it's there and it starts to respond to that tumor that's there. And that response may also be interesting and important for us to find as a detection biomarker. And Lynn mentioned this in the relationship with diabetes, right? In that case, that's really what's happening. The body is reacting to the tumor that's in place and people's blood sugar is rising. And so there's really a whole set of studies around these same ideas. Many of these are using things called metabolites, which are small little molecules that are the products of your body's use of energy and nutrients. And glucose is a metabolite. That's one of now many that are being investigated to try to say, let's not only leverage what the tumor releases into the blood, but let's actually also leverage what the body is doing in reaction to the tumor to try to find it early. So again, really using as much of our knowledge as we can of an early cancer to put together these different markers and try to find the cancer early.
Great. Thanks, Brian. That was really so interesting. It's amazing how complex our bodies are and all these things that we do to try to figure out what's happening in there. So Rosie, one of the things we hear a lot about some of these tests that can detect up to 50 cancers, including pancreatic cancer, called PAN meaning across all cancers, pan-cancer tests. What are those all about?
Yeah, thank you, Julie, for the question. It is, so far we've been discussing this idea of pancreatic cancer really needing for any kind of screening to really focus on a high risk population. These pan-cancer tests, so to speak, are really looking towards being able, like, how could we come up with a test to screen the general public? And so these types of pan-cancer detections where they can detect many different cancer types, they have an eye towards being able to put this into a clinical setting where it would be something that every time you went into your doctor's office for a physical, for example, you could get this test. And to make something that would work like that, I'm going to bring up a couple principles that Brian really beautifully described. And that is the idea that researchers have been trying to capitalize on, which is that tumor cells are often, because they're dividing more frequently and their DNA is what we call kind of unstable because they're not well-controlled normal cells. They are constantly dying. So even though they're growing and dividing, they're dying too. And this causes the release of tumor-specific DNA into the blood. And this is called cell-free DNA that the scientists can measure. But as Brian also mentioned, one of the important things about that is that all of the DNA in your body, depending upon which cells they are, will have different marks on them, these methylation marks. And so scientists have been able to kind of capitalize on that. Like if you can imagine that all the cells in your body have the same DNA, but the cells that make up your heart, some of that DNA needs to be used to make heart cells, whereas the cells that make up your pancreas use different sets of genes. And so the body or the DNA is marked with these methylation marks to say, okay, I'm a heart cell or I'm a pancreas cell. And the idea with these pan-cancer tests is that they would not only look for cancer-specific mutations that would be in the blood, circulating in the blood, but they will look at these methylation marks, and that will then tell them, okay, there's mutations that indicate maybe this patient has cancer somewhere in their body, but now we look at these methylation marks and we can say, oh, those are coming from cells of the breast or of the pancreas or of the liver. And so really the tests that are coming online now and that are actually in big, large clinical studies, they're usually multi-analyte, meaning they're combining several markers, and they're mostly based also on having these methylation marks. So, for example, two of them that are in ongoing clinical trials, one of them is called CancerSeek, which is being developed by Exact Sciences currently. And CancerSeek uses proteins, also looks at DNA mutations, but also is now adding in DNA methylation markers to come up with a multi-analyte test that would be pan-cancer. And there's large clinical studies with 10,000 patients being enrolled in to test the validity of the CancerSeek test. And it's looking very promising. In general, the ability to detect where the cancer is in terms of what the cell type is has like a 99% specificity. It's very high. The sensitivity, the ability to see that there's a cancer in the patient depends upon the type of cancer. I think for pancreatic cancer, actually, the CancerSeek is at about 90%, which is very exciting. Grail is another company, and they have their test called Galleria. And that test also is actually strictly based on DNA methylation. And this, again, they're enrolling large numbers of patients. Our center is actually involved in the Pathfinder study, which is enrolling 6,200 patients. And again, because the idea with these pan-cancer studies is that they would be able to screen the population in general. To be part of this Pathfinder study, you only need to be 50 years of age or older. And then you can be asked by your doctor, do you want to participate? And if you do, they are stratifying patients who are at risk for cancer because of genetic mutations or other, but then there's also patients who have no reason to be at risk for cancer. So all 6,200 patients are if you're over 50, are being enrolled. We've so far enrolled 1,700 patients. And the nice thing about this test is the data is being reported in real time back to your physician. And it's going to be all compiled in a large study to see if there is an advantage to this as a general public screening tool for cancer.
Great, really fascinating. And thank you, talking us through some of the studies that are being done to test and validate some of these tests that we've been talking about. So usually the first step in validating a test in a study is a retrospective study, meaning the researchers go back and look at a bunch of samples and clinical data from people in the past to try to draw conclusions about how a test might work. And then at that point, they do a prospective study to prove that hypothesis. So maybe, Brian, can you talk about a study like this that you're working on?
Yeah, sure. Thanks, Julie. So I think the first point is, as Julie's alluding to, it is a long multi-step process to go from the initial lab work that suggests, oh, this is a biomarker that might be interesting, all the way to a clinical study that proves that. It's really a many-year and large effort to do that. And along the way, there are a lot of different steps, right? It really starts in the lab with a lot of lab work to try to understand what a good biomarker might even be. Then, as Julie is suggesting, then it's a study saying, well, these individuals have cancer of the pancreas with blood or some sample drawn at the time of their diagnosis, and let's compare it to a general population group that doesn't. Lynn mentioned this at the beginning too. Then often another step is to ask, well, what if you had blood samples drawn before diagnosis, right? And let's see, if you go back 6 months or 12 months or 24 months before a patient would have normally presented because of symptoms, how does your biomarker work then? and then ultimately to a prospective study where you actually intervene, right? You then do something different if the results suggest that a cancer may be present. So in terms of thinking of studies that we do in this area, there are many. I thought what I would highlight is something my patients often ask me, which is they often ask, do you collaborate with other centers on this work? And I thought I would give an example that really suggests and truly is the answer, which is yes, extensively, right? We really work together across the entire country with other investigators. And so the projects that I thought I would highlight are through a consortium based at the National Cancer Institute called the PCDC, which is the Pancreatic Cancer Detection Consortium. And this is a consortium of 8 different teams of investigators around the country. Within each team is often many different institutions also. And then all of us across these 8 teams and these many different centers within those teams all work together. And we're really all working together for the exact purpose that Julie was suggesting, which is to design studies like this that can more rapidly move biomarkers from the lab ultimately to prospective and then ultimately interventional studies. So an example of the retrospective samples where we have blood samples from the time of diagnosis and we have samples, meaning at the time of diagnosis in patients who have pancreatic cancer, and then the general population, is something we're calling our biomarker bake-off study. And it's a study across all of the PCDC where 3 of the sites are all contributing samples. Dana-Farber, where I work, is one of them. And then 5 different labs are all getting those samples in a blinded way, and they're doing their biomarker or markers measurements on those samples. And then all the data comes back centrally, and we really try to parse through and say, if this is a bake-off, not all the things we bake are going to work well, and some will, right? And which are the ones that really are working, and which are the ones where the recipes should be retired, right? And so we're just in the midst of that kind of big study. And we really, as this consortium, have felt there's a lot of good and promising things out there, but how do you really compare them one to the other? And how do you make sure that these are really the ones to then invest a lot more time and energy in to develop further? And this bake-off approach is one way that we're doing that. And that approach has a host of different kinds of markers, proteins and DNA and RNA. Again, we really want to understand how these different biomarkers interact with one another. And then I'll give you an example also of trying to find samples in patients or have samples from patients before they're diagnosed. That's really quite hard, right? There's not a lot of resources out there like that. And I think Lynn mentioned there are some studies where people are enrolled to a study, they give a blood sample and then they're followed over time. But there aren't that many of those studies around. And most of those studies are not pancreatic cancer specific, right? They're for the general population. And so it means the number of pancreatic cancers that develop, fortunately, is not that high. So what the consortium has done, this PCDC, is to, across all these different sites, so again, many sites around the country and actually now expanded around the world, are asking patients that are either at genetic risk, so what we've talked about before with genetic mutations or family histories of pancreatic cancer, or actually what Rosie was talking about, so these little cystic lesions in the pancreas, are they willing to follow with us and give us a blood sample every 6 to 12 months over time? And I think as the people on this call know, many, many patients are very generous with doing these sorts of things. And they're very willing to do this if we think it can help find the cancer early in people down the road. And so all these centers have now come together. This is called our signature projects for this consortium, where we're now drawing blood samples every 6 to 12 months from these very kind patients, with the idea that as we go through our bake-offs, right, and these other centers work to find the right biomarkers, we then can start to understand when in time you can find them before the clinical diagnosis would have occurred. And that's really going to be important to us as we ultimately design screening protocols. You do need to know how frequently should you be testing these things and how far in advance are you going to find these biomarkers. And so those are a couple examples, to Julie's question about what are we doing in this area? And again, just to really tell people, there's a tremendous amount of collaboration in the pancreatic cancer research community to come together and do this work together so we can most rapidly advance these biomarkers to the clinic.
Great. Thanks, Brian. I love the bake-off concept. That's great. And I think such a good point that this work really has to be done in collaboration. It's complicated. There are a lot of steps. It requires different disciplines. And maybe most importantly, it usually requires quite a high volume of patients to enroll in these studies in order to really validate these tests. So it really it would be hard for one institution to do any of these things by themselves. So, Lynn, as Brian just said, there are not as many interventional early detection studies. This means that the study is being done in real time with the opportunity to identify patients before they're actually diagnosed with their pancreatic cancer, rather than studying them after the fact. PanCAN's early detection initiative that was launched yesterday is an interventional study. Can you tell us about the early detection initiative?
I'd love to tell you about the Early Detection Initiative. And let me start at the beginning, which is the Recalcitrant Cancer Research Act. And the devoted volunteers and advocates, many of you that are in the audience, I suspect, went to Capitol Hill and told the government that we needed more research in pancreatic cancer. The National Cancer Institute responded to that mandate from Congress. And came up with a strategic plan that had 4 priorities. And one of those priorities was, let's understand the interaction between pancreatic cancer and diabetes. And that resulted in a study that's called the NOD study, the New Onset Diabetes Study, that is done actually through 2 institutes within the National Institutes of Health. So a collaboration even among government agencies. That got this study going. And what they do is they go across the nation. They look for individuals whose blood sugar just went over that level, that threshold that we call diabetes, just recently. And they did not have evidence of an elevated blood sugar 12 to 18 months before that. So it's new onset diabetes. And what they're doing is they're collecting blood from these individuals and they're storing it, and they'll be able to do these kinds of bake-offs and studies looking at the various biomarkers within this population. And as we said, new onset diabetes has an elevated risk for pancreatic cancer. It's a 1% risk in that group. So there will be cases of pancreatic cancer that will be detected within 3 years of that elevation of blood sugar. And so we'll be able to use those samples. So what PanCAN said was, that's great. We really want, that's going to be a fabulous resource for this. But let's see if we would do an image at the time of new onset diabetes. Would we find pancreatic cancer at a much earlier stage? Would we find it at a time when that individual was eligible for surgery? Would we be able to catch it so that that individual would have a lot more opportunities for therapy that would improve survival? So PanCAN it's a separate study, but it's very much connected to this new onset diabetes study that the NCI is running. It'll be done in some of the same places. We will use the electronic health records to find those individuals with new onset diabetes, elevated blood sugar. And then we're going to look at a subgroup of those that will look at how fast that blood sugar is changing. We'll look at changes in weight, and we'll look at age as well. Everybody will be over 50, but how much they're over 50 will make a difference. And we'll take that subgroup of individuals and we'll image them with CT imaging. And we'll ask whether we can find pancreatic cancer with one, and if not one, with a second image. And we'll be able to then follow those individuals, and we'll actually determine whether imaging at the time of new onset diabetes is actually a protocol that we can use across the nation to find some of those individuals with pancreatic cancer at a much earlier stage. So it's a randomized, controlled, interventional trial that will give us an answer that we can use to determine whether this is an approach to the early detection of pancreatic cancer.
Great. Thanks, Lynn. We're really excited about it and honored to be running this study and contributing to the field in this way. So this has been a terrific discussion. We have a lot of viewers on our call today, and they're sending in questions. So I want to turn to a few of the questions that have come in from our viewers. So Brian, kind of going back to the basics, can you explain the ways in which pancreatic cancer is currently most commonly diagnosed?
Sure. Thanks, Julie. And also just a quick congratulations to PanCAN for getting the early detection initiative started. It's just a tremendous effort. It's going to be very important for the field for us to learn from that. So big congratulations is due. So if we've obviously been talking a lot about early detection and how we can find it early, but we certainly can go back and ask, well, what happens now? Right? How are patients primarily being diagnosed currently? So I would say when patients come to clinic, most often it's been one of three things that's led to their diagnosis. One is they have some non-specific symptoms. They have, say, some abdominal discomfort, or they lost a little weight, or they just weren't feeling right, and they told their doctor and it just continued. And ultimately they got some sort of imaging study to find the pancreas. Could be ultrasound, could be a CT, but it's often a long process because the symptoms are not very specific. And so it can be hard for a primary care doctor to know, oh, these are symptoms of pancreatic cancer. And it can lead actually to quite a bit of time going by from when those first symptoms start to when the cancer is diagnosed. Because again, these symptoms tend to be relatively nonspecific. So that's one thing. A second that we sometimes see is that patients present with jaundice. Right? So they have a tumor in the pancreas. That tumor happens to sit right next to the bile duct. And the bile duct is what delivers bile from the liver to the small intestine. And when that bile duct gets pinched or blocked, the bile backs up into the liver and ultimately into the bloodstream. And that makes people turn yellow, right? That's what jaundice is, where they turn yellow. People will often also have some changes in their stool and urine because of where the bilirubin is going. So that's another way. Sometimes people will say, oh, I don't look the same color. I look yellowy compared to what I did before. And that usually makes people go to their doctor. And then that ultimately leads to the diagnosis. A third way, so first was some nonspecific symptoms. Second is jaundice. A third common way that we sometimes see, although I guess I would say it's a little less common than the other two, is they had an imaging study for some other reason, right? They were in a car accident. They had appendicitis or something else, and they had a scan done, and that sees the cancer when they really didn't present with symptoms specifically. It was found incidentally is often how it's described. But I think as you might be able to tell from these things, again, there's a long time off, and particularly for the most common way that this cancer is diagnosed with these nonspecific symptoms, from the initiation of those symptoms to ultimately the cancer diagnosis. And there really is a lot of room here for us to try to improve how we diagnose this disease. And even better, which has really been the topic of this conversation, can we do it before symptoms develop? Because often once the symptoms develop, it means the cancer is starting to spread. And we'd really like to avoid that, right? We'd like to find it before there's any spread. And that really means we need to focus our attention also on the time frame before symptoms develop.
Great. Yeah, we certainly don't want the only way to be able to detect pancreatic cancer early is because you're going in for surgery for something else and they discover it. So it is exciting to think about the possibility of actually having an early detection test. So, Rosie, we see these commercials all the time for Cologuard, for colon cancer, and you have to provide stool. So this question from a viewer is, are there any promising advances related to early detection for pancreatic cancer through stool samples?
Yeah, well, I want to thank the submitter for the interesting question. I think it is a really interesting, exciting area. And surprisingly, all the things that we've discussed about blood markers like proteins and DNA that are released from tumors can actually be detected in stool samples. And that's part of the Cologuard test. There's some of the tests that have looked at that in pancreatic cancer. It's definitely much earlier and there's much less work on it than the blood markers. And in general, I would say what's been found is that while the specificity is very high, so if you showed, mutations or DNA markers or protein markers, it can have a high specificity of detecting the pancreatic cancer, but the sensitivity is pretty low. And I think that there's sample collection issues and such that could be affecting that. So I think this future will tell whether a fecal test could be in play for pancreatic cancer. Right now, most of the work is on liquid biopsies, blood biopsies.
Great, thank you. Brian, so you talked about, some of the common ways that we diagnose pancreatic cancer. What about reoccurrences? How do we diagnose those? And is anything that we're sort of talking today related to, how we might detect reoccurrences?
Sure. So it definitely is related in that setting too. So in terms of recurrence, generally that means someone had a localized tumor in the pancreas. They underwent surgery, so that tumor was removed. And now we're following patients after their surgery, hoping obviously that the cancer doesn't come back with all the aggressive treatment that they had, but looking to surveil them. So have a surveillance program to check if the cancer does. Because if the cancer does come back, we'd like to find it on the early side, because then we can institute treatments for them earlier, right? When treatments may be more effective. So right now, if you ask in the clinic, what are the main things we do for surveillance of patients who've undergone surgery and we don't see the cancer anymore because it's been treated? It's a combination of blood tests and imaging studies, in addition to sort of old-fashioned doctor things like histories and exams and things, which of course we do and can be useful. So it's really those three things that we use primarily. So history, physical exam, blood test, and scans. In terms of blood tests, right now, the primary blood tests that we use are some of the tumor markers that have been around for a long time. So we mentioned CA19-9 or CA19-9, which is a protein marker that the tumor can make and ends up in the bloodstream. We use that to follow patients after their surgery and treatment. And sometimes, in some patients, that can be the first marker that the cancer has returned because that blood level goes up. There's a second marker, protein marker, called CEA that we also sometimes use. That one's a little more variable from sort of doctor to doctor or center to center, whether they use that one or not. Some of us do, and we use those too. In terms of scans, most of the time we do CT scans for follow-up. So usually of the chest and the abdomen and the pelvis at intervals after the surgery and treatment, again, with the goal of trying to find the cancer earlier. However, even though these are some of the standard things which we've been doing for a while, many of the blood tests and approaches we've been talking about also can have relevance here, right? So if you're trying to use a biomarker to find a cancer early, that same biomarker may be also useful to find the cancer early if it returns after a surgery. And there now are studies ongoing trying to use some of these same types of biomarkers, but to use them after surgery. Again, trying to find the cancer earlier after someone has undergone their therapy. So these two areas or sort of fields of study really do overlap with one another and inform one another. And I think as these biomarkers advance, they're likely to have relevance to both early detection of the cancer initially and also looking for recurrence after someone has had definitive treatment.
Great, thank you. So Lynn, Brian, talked about the new national guidelines that require or that recommend genetic testing for all patients. Can you talk a little bit about what companies are doing the most comprehensive genetic testing? How does a patient know what testing they should get?
So yes, those are the guidelines. What normally happens is an individual goes to see a genetic counselor. It's really the genetic counselor that will order the test, and that they will know the companies that are qualified, accredited to do the kind of tests. You want to do a very thorough, deep test for someone with pancreatic cancer to make sure that we look for all those alterations, those genetic alterations that we know is possible to occur in those genes that predispose to cancers like pancreatic cancer. So that's important. And it's important to do it through the genetic counselor. there's also a lot of studies being done that would be great to be involved in these studies. There's genetic registries and there's surveillance programs all across the nation. The best way to find out about them is to call PanCAN. Call our patient services. We have a fact sheet that has about 20 surveillance programs all across the nation, including Dana-Farber in Oregon. And there's genetic registries at those places. Again, some are studies, some are collecting samples that can be used for research in these very important studies that we've been talking about. So there's a lot of information that is available. I think, again, the easiest thing to do is to call PanCAN. We have a list of those sites. We know that those are sites that are doing important work and work that will help not only the patient today, but the patient of the future as well.
Great, thanks, Lynn. So let's take one more question from our viewers. Brian, at what point in time do you recommend that someone who is at a higher risk be screened and or regularly monitored? And what should they do? What's the next step?
Thanks, Julie. So I'd also just reiterate what Lynn was saying, which is genetic counselors are heroes, right? They see patients and really help them assess risk and decide what sort of panels of genetic testing to do. And that's, again, really important, and all patients with a new diagnosis of pancreas cancer should have that done. So I think in terms of when should screening happen, that's a little bit of a complicated question, meaning there's not really one single answer to that question. So there obviously are these different groups of risk that we've talked about. If we think first about the genetic side of things, so genetic risk, there really are a number of factors that determine when that should start, meaning when screening should start. It is which gene is mutated. So it turns out that the different genes that someone can inherit that may be altered don't confer risk in exactly the same way. Some of them confer risk when you're younger. Some confer a higher risk than others. So which gene is actually altered matters in terms of when you start screening. Another is, what's your family history like? So the number of relatives of yours that had pancreatic cancer, that may change when screening is recommended to begin. And then also, what was the age of your relatives when they got pancreatic cancer? So if there's a lot of very young people in your family who got pancreatic cancer, the recommendation may be that you start screening at a younger age than if the family members who got that cancer older, right, when they were older. And it really reiterates a point that I think Lynn was making too, which is you do want to go to a place that knows this literature and knows how to manage patients like this well. This is not so straightforward. It's not sort of a yes, no. There's a lot of gradation here in terms of recommendations. And there are centers that really specialize in counseling families who have pancreatic cancer in their family, or who are families that carry these mutations, about when to start screening and how frequently. And I think another part of Julie's question is, well, how frequently do you do that and when do you stop? Again, these are not simple answers. These really depend in an individualized way on you and your family. So we may recommend screening at shorter intervals, depending upon the findings in your family or the findings from the last scan you had. And it may be longer intervals, depending upon those features. When to stop screening is actually not so easy. We're not really sure when you stop screening. That really is a discussion with the doctor that you're seeing, because it really relates to your other medical problems and the risk of pancreas cancer, depending upon which gene or your family history. And I think if we go back to what Rosie was talking about with cystic lesions, again, it is not simple, right? There's no one answer for everyone who has a cystic lesion of the pancreas. It really does depend on what the characteristics of those cysts are. How are they changing over time? So again, you really do want a clinic and a doctor who know how to deal with these conditions and do this every day as part of their job, because there's a lot of subtlety and differences here. And again, I think the genetic counselors and our genetics teams, particularly for those subsegment with genetic factors related to their risk, play a really important role in helping to craft this sort of plan, both for patients and their families.
great. Terrific. It really is, and I think it's such a good point that it really is so critical for patients to go talk to specialists who are very familiar with this disease. This is not simple. It is complicated. And of course, you can call PanCAN's patient services to learn about specialists that are in your geographic area. And I have to say, you know, as someone who's been following the pancreatic cancer field for over 20 years now, although we still have a lot of work to do, boy, have we made a lot of progress. And I just feel so hopeful and that we are really getting closer to potentially having an early detection test for this disease. So I want to say thank you to our panel. You guys were terrific. Thank you, Dr. Matrisian, Dr. Sears, Dr. Wolpin. Really amazing. And thank you to all of our viewers, our PanCAN family, for trusting us to deliver this really critical, important information to you. We are so optimistic about everything that we heard today, and I am so excited about PanCAN's early detection initiative. Without your support, it wouldn't be possible. So thank you what you're doing to change the future of pancreatic cancer. You can help continue to enable our mission and support all of our research initiatives by visiting us at pancan.org and making a donation. Please also remember that you can contact our patient services program for any information that you need about pancreatic cancer, but also any of the early detection studies that we discussed today. Our case managers are available Monday through Friday from 7 a.m. to 5 p.m. Pacific time. Please make sure to utilize this service. They really provide amazing information so that you can make informed decisions about your care. From all of us at PanCAN, thank you so much for joining us today. Please be safe and stay well.
Thank you for joining us today. All production occurred in accordance with local, state, and federal guidelines to help slow the spread of COVID-19. If you still have a question, please contact patient services. From all of us at PanCAN, please stay safe and be well. Have a great day.